From Microbe TV, this is Immune, episode number 21, recorded on June 11th, 2019. I'm Vincent Draccaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Welcome back.
And from Durham, North Carolina, Steph Langle.
Hello, hello. Sorry, I introduced myself with my giggle before when you said May. My ears perked up.
I could cut it out. I don't know. No. I don't know why I said May.
I like it. I like the little airs.
The June is staring at me right there, but I'm just not.
Time's
processing.
Flying, and we're not ready for it yet.
Just not processing. Before
you guys have, go ahead. Sorry.
No, go ahead. Ask your question.
Well, I was going to ask what your summer looked like. If you had a lot of travel, if you're going to be staying local.
What are you doing, Cindy?
I'm going to go to Germany for 3 weeks.
And what's happening there?
So I'm going to work with a friend of mine, Ruth Lay, for a few days. We're going to strike up some collaborations and go over some data that we have.
Does she have the same name as you, Leifer?
No, Lay. Ruth
Leifer.
Lay, just Lay.
Oh,
no Leifer, just Lay.
You said Leifer before. I'm going to play it back to you.
Oh, okay. Lay.
I thought
Ruth
it was
Lay.
like someone in your family.
No, Ruth Lay. Anyway, she and I have done some work in the past, and we're going to go chat, and then we're going to go play. I'm going to play.
What part of Germany? What part?
In Tübingen. She's at the Max Planck.
Cool.
Yeah.
How about you, Steph? Where are you going?
I'll do a couple family trips. I'm going up to Wisconsin a little bit. I'll go to Ohio for a wedding, but then I'll be around here most of the time. Lots of milking mice.
Where are you from originally? Wisconsin or Ohio?
No, I'm originally from Canton, Ohio, but my husband's from Madison, so we'll go up there.
Huh. Well, that's cool.
Yeah. What about you? Will you travel a lot?
Yeah, a lot. Next week, I'm going to ASM in San Francisco.
Nice.
Then I'm going down to San Diego to do a podcast, and then I'm going to Baltimore to do a podcast all that same week. And then July is ASV in Minneapolis.
Yep. Yeah.
And August I'm going to Sweden for a week to do a podcast. I was invited by the Swedish Society for Virology.
Oh, great.
I'm doing it with a guy named Erling Norby, who's a virologist who has been on the Nobel committees, and we're going to talk about Nobels in virology.
Awesome.
And then in September, I'm going to Copenhagen, and then in October, I'm going to Georgia. I know, Georgia.
Oh, you stop. You're so funny. And then Georgia's a fine state.
And then in November, I'm going to Munich, Germany. And I think December, I'm probably not going anywhere. But hey, if you want to invite me somewhere, I'll go. Just kidding. And I have 3 European trips lined up for next year. It's nice. Cool. It's very
cool.
Have podcast, will travel.
That's good. I like that. You should get a bumper sticker that says that.
Yeah, it would be a good thing, wouldn't it? Anyway, if you like our podcasts, it's a mix of having conversations like this one about papers and going on the road and talking to people. You could support us financially. We would love that. You could go to microbe.tv/contribute. We'll talk about that in more detail later, but I just wanted to give you a heads up. And speaking of heads up, we have a follow-up this week from Amy, who writes, Dear Professors Immune, I just finished listening to the episode 20 about coagulation. It reminded me of the summer that my mother went through repeated cycles of hyper and hypocoagulation that the doctors could never explain, as she suffered from an autoimmune disease, Wegener's granulomatosis. This episode made me question the role of her inflammasome in that and other weird episodes. It illustrated to me the importance of different fields within science and medicine working together instead of each scientist staying in their own bubble. I appreciate all that you do to encourage communication between scientists. What is Wegener's granulomatosis?
So it is a condition that causes inflammation of the blood vessels. So makes sense. And it's, rare. Oh gosh. It can affect any organ, but mainly affects the sinuses, nose, trachea, lungs, and kidneys. I'm trying to see if it's autoimmune. I mean, it says inflammation, but I don't know what target is. Anywho, but that's interesting. And if you think back to our first episode when we talked about the effects of cold stress and temperature dependent responses, that's interesting that she said it happened over the summer. I don't know if that means it does not happen in the winter, but another layer.
Well, thank you, Amy. Here at Immune, we go outside of our bubbles.
We do.
That's right.
In fact,
sometimes far outside.
I would never be speaking.
Foreshadowing, Cindy.
I would never be speaking with Steph or Cindy if not for this podcast, because we're in different fields. Probably not. I wouldn't, right? But Steph, I kind of knew you you used to write. You wrote to TWIV. You were on TWIV, right?
Yeah, there's a crossover with the corona virology and the viruses. But you're right. I think relatively different fields.
But Cindy, I met at a seminar, and probably I would never have spoken to her again after that, except that she's here on Immune. But I think I like this crossover, and I do 5 podcasts. Only one is in my field of virology, but I love it because it's all fascinating. And I think someone told me once, Elio Schechter on TWIM, he said, "You're very brave to do some of these papers that are not in your field." I said, "Well, no, I don't think it's brave. I just love it. Yeah. Very curious." And
I think it's great. You bring a perspective that our audience brings too, because there's probably a lot of people out there that don't know these immunology things. And when you ask questions, they were probably thinking the same thing. So it's harder when you're deep in it to back out and think about the big picture.
But I do think scientists in general need to step out of their own field. And we don't do that very well. So that's why these podcasts are fun for me personally. But, here at my department, we have a seminar every week, and it's very sad because for the immunology speakers, the immunologists go, et cetera. And I just think it's very sad that people don't want to hear outside of their own field. I'm too busy. Well, that's not an excuse. You're a scientist. You should learn everything. So
I love to go to things that are outside because that's where you get these amazing connecting ideas that you wouldn't have thought of otherwise.
For sure.
Right? I think that's the fun part of science. If you just always do your own little thing, gets a little.
And I love when I travel to biology departments, they have people doing all kinds of stuff.
Yep.
Yeah, definitely.
You and I are in micro and immunology departments, the same pretty much. But you go to places that are studying cacti and fish and bugs. It's amazing. And of course, on the same campus, you have physics departments and chemistry and astronomy and all kinds of stuff. And you could go to those seminars too.
I wish every one of those fields had their own podcast. So if you can't travel to their seminars, you could.
Well, some of them do.
Some do, right?
There are plenty of astronomy podcasts out there.
Right. Ecology,
there are, there's plants, there's physics. I don't know.
They should all be on your website so we can easily find them.
Yeah.
Dig right in.
I just wish you could be students, right? Why do we have to work? Can't we just go to seminars?
I once thought that I would like to go to college again.
Oh, yeah. Go back with a totally different perspective.
Now, just go to college and take classes because it was wasted on me when I was young.
Yeah.
Totally wasted. And now I could have, it's not fair to go to college after you've spent most of your life working, right? But I think I would get so much out of it, not just science courses. I would love to take all kinds of courses and feel dumb, like, I don't know what that is.
Yeah, no, I had blinders on, right? I had a mission and I was too focused and I didn't take advantage of all the opportunities that were available when I was in college.
Yes,
right?
For sure.
For sure.
And you don't look at things with the same view. You have a life's view, and now you can go take literature or history. I would love to take history courses, right? History is so cool. Anyway.
But the beauty of the internet now is, though, you can go take a history class online, right?
You could.
You don't have to pay for it.
That's true.
Yeah, you could. But for me, I love to be in class and ask questions of the professor and listen to other students asking questions. I love that. That's the best part. Just being there. I know the internet, makes it possible for anywhere in the world to take courses, and I certainly take advantage of that by putting my lectures online. But man, there's nothing like sitting in a room with people just for an hour and have a conversation and listen to them. I think that's great. I mean, maybe that's just because I'm older, but I do think it's a great way to learn, right?
Yeah.
Yeah, definitely.
Okay.
And we're going to learn today.
Oh, boy.
All right. Are you guys ready?
It's Steph's turn.
Woohoo. All right. Yep. So I'm going to be talking about a paper today that was kind of hot topic. I think it was last week or 2 weeks ago, kind of hit a lot of the major news cycles. It's all over Twitter, and really because of the implications on basic immunology, but then also some implications for type 1 diabetes as well. This is a beastie of a paper. So I'm going to introduce a couple concepts. We're going to ease into it. I'm kind of going to work through what they're trying to convince us of. And then the end, and you guys interrupt me with questions and we'll see what we can do. And there will be parts we may not know ourselves. And if you know, you can write in and tell us where we got it wrong. So I'm first going to start by talking about B and T lymphocytes. So they are the two main lymphocytes of the adaptive immune system. The adaptive immune system happens later. It's specific for a particular pathogen. And in comparison to the innate immune system, that's going to happen within hours of a pathogen's appearance or minutes, as we learned last week for coagulation. B and T cells are going to work together to recognize whatever particular pathogen later, days later, weeks later. And these two cells are defined by two receptors at the cellular surface called the BCR and TCR, or the B cell receptor and T cell receptor. So both of these receptors have similar structures. They're both heterodimers. The BCR, which I'll refer to, the B cell receptor, is made up of two components called the heavy and light chains, whereas the TCR, or T cell receptor, is made up of alpha beta chains. Those are the variable chains, so they're going to change dependent on the pathogen that's presented to them. There are other components of these receptors. They're the invariable components that don't change but are really integral into signaling through those receptors. And so how these receptors are able to recognize the vast amount of pathogens and antigens and things in our bodies and outside our bodies is they're able to rearrange gene segments from a randomly selected pool of unarranged gene segments. And the ones that they're rearranging are called VDJ. V is for variable, D is for diversity, and J is for joining. And so they take from this random pool of unarranged gene segments, and they're going to bring them together and arrange them or recombine them, randomly assemble them to create a region on these receptors. So there's a region that we're going to call CDR, but the name is complementarity determining regions. And you can think of these as like if you hold up three little fingers on each of the chains, each of the heterodimers, these three little fingers, if they at this top of this receptor, if they're close together, they can hold an antigen that's small as it's being presented. If they're wider, they can maybe hold something a little larger that's being presented. But that specificity is what helps them respond in a specific way. And they are highly variable, again, because of all the different antigens that your genes, we just don't have enough genes to encode how many antigens we're going to see in our lifetime. So I think 10 to the 18th is really the massive number that we say that's, kind of repeated over. I'm sure we can try to find the original paper who did the algorithm for that, but you can get the picture is really an unlimited number of antigens. And so the random assembled VDJ gene segments help to accomplish this, in addition to other diversity enhancing mechanisms. And that would be, there's a certain enzyme that can add nucleotides in between the junctions of these VD and DJ segments. And that can also increase the diversity. And diversity is a good thing, like I mentioned, to protect against the host. But when it's a problem is when BCR or TCR are recognizing self-protein, when it's recognizing something that it shouldn't be and having a reaction to it. thinking back to previous episodes we've had where we've talked about control of our B and T cells and positive and negative selection, both in the bone marrow and the thymus where they're from, but also in the periphery. And there are so many different checks and regulations on these cells that allow them to, if they bind too tightly and they recognize a self-protein too strongly, to be selected out of the pool. If they don't bind well enough, they fall out of the pool. And this kind of dynamic keeps things working so that we're binding the things we need to, but not self-proteins. Of course, this does happen a lot, and your genes can determine whether or not you're more prone to recognize self-proteins, but that is what autoimmunity is. And it can happen more in genetically susceptible individuals, which I'll talk a little more about in a bit. So what this paper, the autoimmune disease that they're talking about and the dysregulation of the B and T cells, is all focused on type 1 diabetes. And type 1 diabetes really is a prototypical autoimmune disease because it is insulin that is viewed as the primary autoantigen, so self-antigen, self-peptide that drives the activation of T cells to kill insulin-producing cells. These insulin-producing cells are called beta cells. They're within the islets of Langerhans, within the pancreas. And so if you have a cell killing those cells...
Langelhans.
Langelhans.
No, just kidding. Langelhans. You never heard of Steph Langelhans?
No, I like that. I think that should be our title. The islets of Langelhans.
Sure.
Sure. Why not? Why not? So you can imagine the problem if you have killer T cells that are knocking off your beta cells that produce insulin. It is an autoimmune disease that causes diabetes. As you no longer can produce insulin, then you have very high glucose causing the associated issues. And I mentioned the disease susceptibility in genetically susceptible people, and it really has to do with polymorphisms or variants at a particular position on what we call major histocompatibility complex 2. There's 1 and 2, but MHC is a molecule that presents antigens to B and T cells. And MHC1 is on the surface of all cells. MHC2 is on the surface of those cells that present antigen. So what I'm really presenting here are these main cell types that we're going to be concerned about in this paper: B cells, T cells with their BCR and TCR, and then this MHC2 molecule, which in humans is called HLA. Among species, there's a lot of different names that are specific to that species. So I'll use them interchangeably. I'll try to stick to one. Maybe I'll try to stick to MHC because that seems to be more, triggers a better memory of what these are. And so...
Can I ask you a question?
Sure, go for it.
So what you're saying is that certain cells have on their surface an MHC molecule presenting a piece of insulin, an insulin peptide, right?
Right.
And that is being recognized by T cells, and the T cells are killing the beta cells, right?
Because the beta cells are going to have insulin.
Right. Now, normally we don't do that, right? We don't recognize our own peptides as foreign and do such things, correct?
We will recognize them, but if we bind, if the T or B cells bind too strongly, they'll be selected out of the pool. So I don't think it's that they can't ever recognize it. It's just that they are not positively selected to go into the systemic.
Oh, you're talking about during thymic development, right?
Correct.
Yeah, so there's this process of education where you throw out all the T cells that are self-reactive, right? But sometimes they get through, as in the case of this disease, right?
That's right.
Yep.
So my question is, what are these peptides doing there in the first place? Do cells just put all peptides on the surface?
Well, it's a great question. And the introduction to this paper kind of lays out this problem with insulin. Because when they take the native form of insulin, which I'm assuming is either they've isolated it from humans or maybe they synthetically produced it, it actually does not bind T cell. It does not bind what the MHC complex that would present it. And so I think normally it would not. The way that insulin is produced, that our body is not going to recognize it as foreign. But what they suggest, what the field suggests, that there is post-translational modification of insulin in susceptible individuals with a certain genetic background that process it wrong. And wrongly processing the insulin is what makes it immunogenic. And so there's a particular substitutions that if you you can do in vitro to show that to be true. But the paradox with type 1 diabetes and insulin is they have found which substitutions to make in the native insulin form to make it immunogenic, but they cannot find that immunogenic variant in humans. Right. So
they put some acidic residues in, and they say, there's hundreds of times more potent at being recognized by T cells, right? And so the idea is that you said there must be some post-translational modification that mimics these acidic residues, right?
Yes.
Now, Steph, they call this a mimotope.
That's right.
Can you tell us what a mimotope is?
Well, I think
is that the right way to say it?
I think mimotope. It mimics the protein, but so not 100%, but it produces a reaction, in this case, that's 100 times greater than the native form of the protein, but it's not yet a super agonist, which they're defining as being 1,000 times better at causing a reaction, at binding. So you can think of the mimitope as an intermediate between a super agonist and the native form of the protein.
So the reason we think this is so in type 1 diabetes is because there's linkage between certain mutations in the MHC2 gene in those individuals. So we're thinking, it must be that it's binding insulin peptides, but we can't really find any that bind really tightly in these people, right?
That's what they say that the field struggles with is they've identified, but they just can't find them. And so where are they? And also you have risk alleles, but it doesn't, if you have this particular allele, it doesn't mean you're going to get it. There are people who don't get it. Right. And there are people who do not get type 1 diabetes, but have antibodies against insulin. So it's recognizing insulin, but there's still some checks and balances on those T cells. So they seem to be this disease neutral phenotype, whereas people who have autoantigens and respond are the people who have type 1.
Got it.
So can I see if I'm also getting this right?
Yeah.
What I understood is that this peptide, when it binds to the MHC holds it in such a way that it mimics what insulin looks like. So that when you have anti-insulin antibodies, they actually bind the MHC peptide complex and mimic that.
Yes, thank you. Because you're right. It's not just about the sequence, but it's about the conformation, the tertiary, the quaternary structure of the insulin to make it mimic. You're right. And that's a great point.
So it's something about this particular sequence of MHC that certain individuals have, and the peptide that gets chewed up and the way it binds in the MHC molecule makes it look like it's actually the insulin. And then they generate antibodies or something like this. So the other thing, I wanted to come back to something that Vincent asked, and he asked, so do all of the proteins inside of a cell get chewed up and presented? And that's actually important because if there's no infection, we still have lots of MHC1 molecules on all the cells in the body. But since there's no infection, they're all self-peptides. So we normally have that, but they don't normally cause a problem because there's no insult and inflammation that's around when those are there. So they're not detected as foreign and not driving a response.
So is that part of, so is it just that the machinery, the proteasome, the loading onto MHC, is it always going constantly? And then if you get infected, then the proteins of the whatever's infecting you will get loaded on? Or
yes,
is there some other surveillance that's going on for self?
So there's a couple of things going on. So there's always going to be T cells that are going around, and if they're seeing any peptides that they're specific to that are presented, but there's no stimulus around, so those antigen presenting cells or those cells that have those antigens aren't also presenting sort of an indication that there's an infection around, those T cells actually get quieted down. And so that's a peripheral tolerogenic mechanism. So that happens all the time. I forgot what, the other thing is yes, there's constant processing of anything that's inside a cell, and it can be loaded onto MHC. And yes, there is in fact some changes that happen when there is a stimulus. So there can be changes in the proteasome, so they chew things up slightly differently. And there's a little bit more effective presentation. So more MHC molecules on the surface, a little bit more efficient processing and presentation. But generally, that whole process is going all the time.
So if you had like a tumor with some different antigens, they would then be recognized, right? They would be.
So that's a tricky one, because if there's no other inflammatory driver around, they might be they would be recognized as self and tolerize the T cells. So that's a way that
some type of neoantigen or something that looked different. It wouldn't, and I think that's why cancer is so challenging.
But that's why the checkpoint inhibitor therapy works, because if you have those T cells around but they're quieted down, if you then remove the brakes, then they can go and kill.
Okay.
Right. Sorry, we digress, but I think it's an important way of thinking about things. And
definitely. So then one of the concepts that they talk about in the paper is something called a clonotype. And so I just want to talk a little bit about clonally expanding BRT cells. And what a clonotype is you have a BRT cell that carries the same unique BCR or TCR rearrangement. And so these designated sequences of B and T cells are recombined variable regions are going to, if they're a part of the same clonotype, then they're going to be of the same B cell or T cell variable joining. And there's different types of segments. So the heavy chain and then the kappa and lambda light chain gene segments. And they're also going to possess identical amino acids in this complementary determining region, or CDR3. I had mentioned these three little fingers that are being held up, 1, 2, and 3. But the third one is the most variable, and it is the most important in terms of detecting a variety of peptides. And so if you have B or T cells that sequences are the same, they're going to be considered clonotypes. And that is something important with this paper. And you can have more than one clonotype. It's just those B and T cells can expand clonally, and then they're able to expand in that way. We also have memory cells that once they're expanded, that they would be all considered one clonotype if they have the same sequences. And so considering the differences between people and how many different types of unique TCR and BCR recombinants that we have to produce, you would think it would be rare that two individuals would actually have the same TCR or BCR sequences. But in fact, that does happen, and it actually happens more commonly than maybe you would think. And it's really because of two different things. One is called convergent recombination, one is called convergent selection. And so I introduced these just because it is talked about in this paper. And it's basically the convergent recombination is when you have biases in the gene segments of VDJ as it recombines. And so there are going to be certain genes that recombine at a slightly greater frequency than others. And so that can drive sequences that occur more likely than others. And convergent selection is more response to like a common antigen. So a virus-specific TCR may happen because a preferential use of a specific variable joining gene in response to an epitope on the virus. So like if there's a conserved epitope on the virus, that would maybe drive it towards having these identical TCR and BCRs. And they call those public TCR and BCRs when it happens in multiple individuals. When you have a TCR or BCR within yourself that's unique to yourself, that's called private. And that distinction is important in this paper because if you can identify public TCR, BCR sequences, you could have a potential way to target with technologies either for, vaccination, for disease where you're targeting like epitope structure-based vaccine design. But for autoimmune disease or cancer, you could maybe recognize things as biomarkers or ways to develop therapeutics if you have a common TCR or BCR amongst multiple people who have this one disease. And so that's important for type 1 diabetes because we don't really know what the peptide is that's going to make this really strong interaction to cause the dysregulation and autoimmunity. And if you can identify these public BCR, TCRs that could be helpful for the things I mentioned, biomarkers and then therapeutics. So the title of the paper that we're going to talk about today is A Public BCR Presents a Unique Dual Receptor Expressing Lymphocyte from Type 1 Diabetes Patients, and it encodes a potent T-cell autoantigen. And the authors on this, I think it's out of Johns Hopkins, and the first author is Ahmed, and the last author is Hamad. I won't go through all the names, but you can look that up. We'll post it in our show notes.
So one of them is here at Columbia.
Yes, the second author.
Second author. Let's see. The corresponding author. One of the two is here. And I also know one of the Hopkins people, Steve Desiderio.
Oh, yep. Mm-hmm.
He was a postdoc at the Baltimore lab at the time that I was there.
Nice.
Oh, cool.
Small
Very
world.
Cool.
Yeah, it is a
very
small
world. Cool. So,
and this paper was published in Cell just a couple days ago, May 30th online, right?
Mm-hmm. Yeah, May 30th.
Now, Steph, you said it was hot. What do you mean by that?
Well, it got a lot of coverage and kind of like scientific media, and it was a lot of conversation on different social media sites as to what the implications are and what this means for the field. So,
okay,
I think that the big thing is that what we know and what has been dogma, absolutely indisputable dogma, is that you've got T cells and you've got B cells. And T cells are T cells because they express T cell receptor, and B cells are B cells because they express B cell receptor. And these are saying that's not entirely true.
Right. Yeah,
that's the big thing.
Yes, that is the big thing. I think, what we're going to go into and what they discover in relation to type 1 diabetes and this T-cell autoantigen, I think that is, separate from maybe what the other implication is that the dogma for the development of these two types of lymphocytes may be wrong. And so we're going to talk a little bit more about that. This paper really sets up and starts out with defining this subset of B cells. And this group had looked at this certain type of B cell subset called CD5 positive, CD19 positive cells. They had looked at these previously and found that in type 1 diabetes patients, that this rare B cell subtype was upregulated in this patient population. And so I'm imagining, I think that paper was published in 2017. And so over the last 2 years had really honed in on this cell type as being unique to type 1 diabetic patients. And so what does this cell do? Let's investigate it further. And, I mentioned these markers on the surface. The CD19 is expressed on all B cells, but in combination with CD5, it's just really a rare B cell subtype, and should not express TCR. And so they really start this paper by telling us that this rare B cell subtype does express TCR. And so kind of makes you go, well, that's odd. It's definitely not something that you would expect to read. And so they're really setting up the paper to prove to it. Yes.
Would they look for that or would they stumble onto it?
I imagine that in investigating the cell type, they're doing single cell sequencing. They're investigating the cell type and saw that.
I see. Yeah.
And that led them down the path, if I had to guess. That's how that went down.
Because these are, even in diabetic patients, these are pretty rare, right?
Yeah.
Yeah. Well, so it's interesting. They're rare in healthy individuals, but they do actually increase in diabetic patients. And this was even discovered in the 90s in type 1 diabetic children. But this cell type increases in a lot of different types of dysregulated B cell lymphomas. Mm-hmm. So I think the upregulation of this cell type is pretty well known within autoimmune diseases and particular types of lymphomas. But the TCR expression is the unique part that sends them down this, tunnel of trying to understand what these B cells are up to. And so,
and I think it's probably something for years people would ignore as
yes.
right?
Yeah.
So if you're doing flow cytometry and you have a teeny tiny population that seems to like a couple of speckles fall in, and they stain with both TCR and BCR, you say, oh, that's just the background, right? Because cells express either one or the other. And I mean, there's selection. This just doesn't happen.
Right.
But now we have the technology, and you're going to go into this, the single cell sequencing, where you can tell in the genetic, on the transcriptional level that they are transcribing both of these.
Right.
And so that can only happen if you, and we could talk about this, if you have issues with your single cell sequencing analysis and it's not single cells, or there is in fact a cell expressing both receptors. And so one can imagine if you're staining with antibodies, you can have a few spurious accidental bindings or whatever. And so you can have this kind of rare couple of cells pop up that might stain with both, but it's background. But if you're looking at the transcriptional level, it's harder to argue that that's an accident, right?
Right. And, exactly, like our dogmas can maybe put blinders on us for things like in this, if you have a population of cells expressing TCR, you're like, oh, that's background. But they dove into it further. And when we're talking about single cells, I mean, they're not talking about a lot of cells and that they're pulling out of these people. I mean, we're talking 4 cells, 7 cells. And so they're really setting up to convince us that the cell type is real. And then furthermore, what does it do? And so they start by looking at flow cytometry. They're saying, okay, it expresses TCR, but it also expresses IgD, which is a surface immunoglobulin expressed on immature B cells, but it can also not express IgD and even express IgA and IgG, which would suggest class switching of this rare B cell subtype. And going into what Cindy said, single cell technology, they kind of approach it in a 3-pronged way. They say, okay, let's look at single cell RNA sequencing, single cell imaging by fax sorting, or single cell BCR assembly. And you can use bioinformatics to look at the sequences and see what that B cell receptor, how it is assembling. And so by going through this, I mean, just looking at the first kind of heat map they have, they looked at single cell RNA sequencing. And I actually should go back and kind of talk about single cell a little bit because it's a pretty hot technology right now. It's really a nice way to tease out the heterogeneity within a single tissue, or, finer resolution than we really could look at before. Before you were pulling cells, you were isolating PBMCs and isolating B cells, but you were looking at RNA in all of those B cells when you're doing transcriptomics. So this allows you to pull out a single cell and say, okay, what is that doing and how is it different from the B cells around it?
So you use a cell sorter to get single cells, right?
Yeah, you can use cell sorter, and there's a bunch of different ways that you can do that. I mentioned flow activated cell sorting, but there's microfluidics, and there's a bunch of different emerging technologies that help isolate one cell at a time, which, as you can imagine, would be really important if you're trying to define rare cell subtypes. Do you guys have any experience?
In some ways no, I haven't done it, but I think some of the ways they do this is you have barcodes coming in one channel and cells coming in another channel, and you put them into a little droplet. And so you have so basically the gene sequences, all of the transcription sequences from that one particular cell get this barcode attached to it. So you can identify which ones come from which cells. So then if you sequence everything, you can assemble it into all the cells had the same, basically like zip code marker. And so each cell has its own zip code marker. And so all the transcripts from that cell have the same zip code. So then you can reassemble to find out which cells were expressing what. And then you can use heuristic analysis to allow it to self-sort into populations based on sequences and things.
Right. So they do that, and they look at the transcriptomes of these cells. So they call the ones that express BCR and TCR dual expressors, or DEs, and they're comparing them to conventional B cells and conventional T cells. And they really highlight the fact that these double expressors do have expression of B cell and T cell lineage markers. And lineage markers are going to refer to either a surface protein or an internal protein or a transcript that's really defining that immune cell subtype that was important in the development of that cell from a stem cell to a mature cell. And so by looking at lineage markers, it's the beginning of telling a story of, okay, this cell it's looking like it's both a B and T cell. But one thing about this that I found was interesting that, they don't really talk about throughout the paper. Yes, it does express B and T cell markers, but it also expresses mast cell markers, monocyte genes, and NK cell genes, which when you look at the paper, the list of those genes in figure 2C is quite small. You really can't make out what you'd have to go to the supplemental to be able to tell. I think that's interesting because it suggests that the cell type, yes, it expresses BCR and TCRs, but it is weird in other ways. It's also expressing these other cell-associated markers. So I think that that's something to keep in mind. And we know single cell analysis, it can be noisy, and those genes are none of those genes are absolutely restricted to never be expressed in B and T cells, but it's just to see that level of breadth, it's suggestive of potentially further issues in these cells.
Are these actually lymphocytes then?
Well, they're suggested. So,
or are they just a tumor cell that's making everything?
Good question.
It's a great question. I think they don't really address that in this paper. They really lean on the fact that they express BCR and TCR. And as I'll discuss in a second, it's on the surface, it's a transcript. So it's a lymphocyte. And I think we could take a step back. And if we were maybe to investigate these other transcripts that they find, could maybe tell a different story. But for moving on with the paper, I think, I'm not as convinced by the RNA-seq data, but let's move on. They did the imaging and they saw the expression of these on the surface. Now, they use the single-cell imaging technology, which is pretty cool to see in the paper. They don't do any double stains, which I would have liked to see because all of these are representative of one cell. So, if they're supposed to express BCR and TCR, it would have been nice to see some double-stained imaging.
But that is it, right? So they're showing you each channel for that one cell. They have a T committed cell and it expresses TCR and not IgD or IgM. They have the B cell that expresses IgD and IgM but not TCR, and then they have their dual expressors that express TCR and IgD and IgM.
I guess that when you look at the supplemental, it's just a little confusing. I'm assuming they've stained. So there's a lot more markers they look at than just in the main paper figures.
Yes.
And I'm assuming that's the same cell. It just, I guess, I don't know. It's suggestive that maybe it's a different experiment. So I was just wondering.
So that the Amnis technology is basically a combination between flow and microscopy. And so you can visualize individual cells through this flow cytometer imaging thing and take pictures of them like a microscope. But you can get all the channels just like you do for flow.
Okay. So even though they're not including that, we can assume that the supplemental and then this main figure, is the same cell.
And these are all cells from patients because you can't grow these double positives.
That's right.
Culture,
that is a catch, right? So they had so few in a healthy individual that they couldn't really analyze them.
Right. And they were able to find these in type 1 diabetes patients. We're going to talk a little bit about if they found them in healthy controls. But okay, so yes, the imaging. And then the last single cell thing they did was they were able to use bioinformatics software to kind of reconstruct the BCR and TCR genes and use the conventional B and T cells as control. And they were able to detect the contigs in both the dual expressors and the conventional B and T cells. So furthering the suggestion that these cells are real and that these receptors, as they try to convince us in the next part, are functional. I think the functionality of these receptors is what now they're interested in telling us about. So to be able to see if they are functional, we would want to stimulate them with a particular, so they use CD3, CD28, which is going to stimulate the T cell receptor. And, after stimulation, you would expect them to be able to analyze the TCR for the cell for upregulation of activation markers or proliferation, but they did not have enough cells because it's such a rare cell type to be able to culture them by themselves. So they had to use general PBMCs, which is peripheral mononuclear cells from the peripheral blood, and they actually cultured them quite long. They cultured them from 7 days, and so this, when they're doing this TCR stimulation, it's an indirect effect, and they don't actually really account for the potential. You're stimulating all cells in that peripheral blood, so you're stimulating conventional T cells as well as maybe these dual expressors. And so, I don't know how convincing this section of the papers for if this TCR is functional. But they do try to go into, describing, yes, activation is upregulated on the dual expressors, the MHC molecules are upregulated. And so then they say, the TCR is functional.
So they stimulate the bulk culture, but then they do single cell analysis, right?
Yeah, they do single cell analysis to show on these dual expressors that they have upregulation of activation and they do proliferate, but you can't rule out that was cytokine secretion from the conventional T cells, which can proliferate other T cells. So it's not a direct way of confirming that. And so then they also look at the responsiveness of looking at the BCR, and they stimulate that by using IgM, and the IgM can help cross-link this certain receptor called CD9, and they use as their readout CD9 phosphorylation to compare with a conventional B cell, and showed, if you stimulate with IgM, it's a functional B cell receptor on the surface of these dual expressors. So this set of experiments is to try to prove to us, the transcripts are there. Is it functional? I think the B cell data is a little bit more convincing. I'm not so convinced about the T cell data.
Why does it matter if they're functional?
Well, I think what they're trying to suggest is that this is a lymphocyte that has an ability to respond like a T cell, but produce antibodies like a B cell. Mm-hmm. Whereas I think it's an aberrant B cell that expresses TCR transcripts. I mean, that maybe was my big payoff for the end, but that's what they're trying to convince us of. And I think that, the TCR functionality is important to prove that.
Yeah, because if these were non-functional, then they would be irrelevant in human disease, I guess, right?
That's right. But they're arguing that these are pathogenic in type 1 diabetes.
Yeah. I mean, but if they're B cells, they still could be pathogenic if they produce antibodies against this, we'll discuss like this potential peptide that causes autoimmunity. But So I have a question for you, because you said you weren't so convinced by the T cell data. But if you think about, when we look at things in vitro, we get very critical of, it's a mixture of cell populations, and you can't guarantee that specific population is the one that's proliferating specifically to that anti-CD3 and a CD28 stimulus. But in the end, if you have all of those cells present in the body, if some of them are responding and secreting cytokine, and that's enough to drive the proliferation and activation of these rare cells, is that enough? Is that convincing enough to say that during a normal body, being present in the normal body, they could be driving this, even if they're not directly triggered? Sure. No, I actually think that that's very possible. And I think you're right. I think that though answers a different question that they're trying to address.
Agreed.
I think that if you're trying to convince me that this is a paradigm shifting lymphocyte, that you would have to have a functional TCR and not have it just be like a bystander effect from cytokines. But you're right. For driving pathogenesis, totally agree.
I agree.
Yeah. So, their next step then is they wanted to look at the repertoire of the immunoglobulin heavy gene, the variable region of these dual expressor cells. And they do this, they compare them to conventional B cells within these three type 1 diabetes patients. And they find that there is a certain gene, and they name it as it's the variable of the heavy chain O4B. And this particular gene is really dominant in this population of dual expressors. And it really is different from the conventional B cells, where they do not use this gene at all, or just minorly compared to the dual expressor repertoire. And so they're trying to demonstrate that this is a unique, it's a unique cell in the way that it uses the different genes through the immunoglobulin receptor. So they're thinking about, well, if all these dual expressors are using this one heavy variable region gene, are they the same clonotype? Because that would be important for determining if this is what I had talked about before, a public clonotype, or is it only specific to this one individual and really can't be applied to multiple different individuals? And so when they looked at these dual expressors using this particular gene, they were the same variable diversity joining segments, and they used the same nucleotides in between the junctions within each of the 3 subjects. In addition to their CDR3, which is one of those little fingers I talked about on the T-cell receptor, were identical, both the nucleotides, the amino acids. So what they're saying is that this is a single clonotype. So these dual expressors are producing the same antibody. Their BCR is the same sequence. And that's important.
Pretty wild.
Which is pretty wild because this is the first time that that's been discovered from in terms of having a public clonotype in these dual expressors. And so they refer to this as the X clonotype because they don't really know what to name these cells. I mean, they say dual expressors, but this X clonotype and the dominance in these unrelated patients, they say is unlikely to be a coincidence because of just how diverse the BCR repertoire is. I mean, that's a pretty amazing finding.
How many cells per patient are we talking about here? Just a couple, right?
Just a couple.
So in all of them, from every patient, they had this weird clonotype, this rare clonotype. Is that right?
I think 3 out of 6. I think there were some that did not,
okay.
But the 3 that they pulled these dual expressors from had the same clonotype.
And why did you call it public?
So public is because it's the same BCR or TCR sequence shared between individuals, which is rare. I mean, it can happen because of the things I mentioned earlier. But if you can identify a public sequence, it can really mean a lot for targeting disease because it's shared amongst.
Okay. So if we compared the BCRs of you and me and Cindy, we probably wouldn't have anyone in common.
Probably not.
Right. Probably not.
So
right. Very few.
Right. And it's such a huge task to do that. I mean, the repertoires of all of our BCR and TCRs. So, you do that by RNA sequencing, right?
Yeah.
Because germline doesn't tell you the diversity, obviously, right?
Well, and so one of the things that they go into is that these clonotypes, they're specific to the dual expressors, but they are of a restricted, so they don't use as many variable genes within the heavy chain so let me go back. They actually were able to find a small population of these clonotypes within the conventional B cells, but they did not use as many gene segments as the dual expressors. And so what they're suggesting is that the lack of diversity, that it's more of a germline configuration. So they did not undergo somatic hypermutation. And so the implications of that are they would be more reactive to self-proteins.
So given a sequence of like this clonotype, and I know you'll get to this, but I just want to ask, can you find out what it's recognizing, what peptide it's recognizing?
Yes. And so when you sequence and you have the amino acids, you can produce a peptide for that core region that binds to the MHC2. And by doing that synthetically, then you can test that peptide, which was encoded by the BCR TCR sequence.
Is that done? Is that done bioinformatically or actually biochemically?
Well, it's bioinformatically first, and then producing the peptide, right?
Got it.
Right.
Okay.
So, can you explain that again? Because I'm not sure that that's totally clear. So it's not a trivial task to go from taking so if you've got the whole immunoglobulin gene, and then you can pick out the part that generates the specificity to the peptide, to whatever it is it binds to. Let's say you have an antibody sequence and you want to know if it binds to flu, right? So if you take that antibody sequence and you use a program to make a 3-dimensional structure, you can see where the CDR3 is. But just from that information, we cannot yet say what that binds to, right? So that has to be empirically determined.
Right. So, I mean, they can then, I'm going through and so, I mean, after they're able to like bioinformatically do that, then they can start trying to pull out the different, using like primers and trying to sequence. I guess I'm not getting what you're moving towards.
So the three-dimensional structure of an antibody, even if you can put that back together from the primary sequence, just looking at what that antibody structure looks like so remember, we think about antibodies as a Y, and the fingertips are what binds to the antigen, right? And so if we know what the sequences are, those little loops that stick out, just because you know those sequences and you can look at the structure of the antibody, we can't predict what it binds to just from that information. I think that's what you were asking, right?
Yeah, because I know they're going to do a more specific experiment later. I was just wondering if you could start from, because they have some hints, right?
Yes.
But if you didn't have any hints and you had this weird clonotype, could you figure out? And I guess
I think it'd be very challenging. Yeah, I think you have to.
I don't think we can do that.
And I think you have to have some previously documented sequence or peptide to move you in the direction, which they do, right? I mean, they have insulin, which was what they're considering is the peptide that this is what we're driving towards, is something that binds better than insulin. So that's what they were using to probe these sequences to then to find what region. So I get what you're saying now, Cindy. I apologize.
I guess it's like saying, if you have a key, can you predict the lock that it goes into, or vice versa? Like if you had a whole pool of them, could you say, oh, I know that goes into that one?
Sure.
And it's not exactly like that, but it's sort of like we can't see what the other side looks like. Right. We can only see what the antibody looks like. We can't see what the antigen looks like that antibody will bind to. We can't predict what that's going to look like.
Right. So yes. And I think because they'll go into that in a little bit, but at least in terms of the dual expressors, and if they're predominated by this particular clonotype, then they can use that information as well as, what we know about the binding of insulin to then move forward to try to understand how, what are the molecular dynamics of this clonotype and what peptide does it produce? And then they're going to start working with that in a little bit. So, okay, I'm going to recap. So I'm going to think about, there's this unique rare B cell subset. They're defining it as expressing both BCR and TCR. They did some functional studies to show it is responsive to, it is that these BCR and TCRs are functional. They're moving on to say, okay, well, what's the repertoire of the BCR and TCR? How diverse is it? Are they similar? And in fact, they are very similar, actually exactly the same clonotypes from the type 1 diabetic patients. And then this dominant clonotype is what they move forward with to try to determine what it's specific for. Okay. So the next section, which I'm not going to fully probably go into, but it's important because they want to know, is it detectable in peripheral blood they just use specific primers, and they're trying to find this clonotype in peripheral blood. And they were able to find it in like half of the type 1 diabetic patients and in some of the healthy controls. But, I think what this is suggesting is it's not in all patients. It's really restricted to type 1 diabetic patients, but also those who have this risk allele. And so, looking for this in a huge population we're going to talk about, they actually fail to do that. So when they look at a public database of this clonotype, they're not able to find it published amongst like 37 million BCR sequences, but they are able to find it in their patients. And so I'd be curious your opinion. Is this so they find it in their patients, but not in this public database because people just haven't found it, or this population is unique and they have dived deeper? It's kind of hard for me to tease out what that means. Okay.
So in their population, they have they have 8 diabetics and 8 healthy controls, right?
Right.
And then they're looking at MHC alleles here specifically, right? And so in you do find it in one of the 2 MHC categories there, right? 3 of 5, but 0 of 3 in the other. And in the diabetics, you get it in one, but not the other. There's only one patient in the DQ7. So
right.
I see it in both, but I don't know why it's not in the database. That's weird.
It's weird. And I don't know if I don't know. It is not. I mean, if they're suggesting that this is totally new, I don't know. Have 37 million BCR sequences is a lot.
Could it be so rare that hadn't been seen before, but yeah, 37 million seems like a lot, right?
And also to find it in 4 of the 8, I mean, they have a pretty good, they should be gamblers it's not in the public, but they're able to.
So Vincent,
yes,
when on TWIV, when you guys covered the XMRV or whatever, and they thought that was, there was some cloning issues going on there, right? So when you're dealing with things where you're amplifying sequences, I guess we have to assume until proven otherwise that all this was done with great care and all of this is correct. But it also could be that somehow this sequence, they're finding it in these multiple things, but it's not actually there in all of them.
Yeah, the XMRV was a clear case of contamination. It was a cell line that actually was contaminated with the virus, and that got into everything. Of course, here they're doing single cell, right?
Yeah.
Yeah, and that's why, I mean, it is convincing. I mean, they have done a good job of convincing me thus far with the single cell that, I mean, it's there, and we're going to talk about the peptide that they produce in a little bit, but yeah, I don't know. It's kind of, so it was just hard for wrap my head around why they didn't find it in any public database. So it may, and they suggest that this may drive type 1 diabetic, diabetes and this type of autoimmunity, but if it's that rare, I'm not sure. So
yeah, because type 1 diabetes is not that rare, right?
Right.
So you would think in those 37 million, I don't know where they come from, those 37 million sequences. Do you have any idea?
No, I can look up.
Depends how many people that comes from, right?
Right.
Because if it's from 10 people, then okay, I can understand this.
Right.
Or even 100. But if it's from tens of thousands or hundreds of thousands, I just don't know. So that's an important consideration.
Yep. So what they did next was they actually produced the protein encoded by the X clonotype, and they call this an X idiotype or XID in the paper. Idiotype, I mean, basically just talking about like a set of binding sites that characterizes the antibody that's produced for this particular clone. So
hold on a second.
Sure.
This 37 million database is from 3 healthy adult donors.
Oh, is that right?
I
was trying to find that. I'm glad you found it.
That's a PLOS One paper from 2016. We present a public database of more than 37 million unique BCR sequences from 3 healthy adult donors. So that would explain it.
That would explain it. So I think,
I'm sorry to interrupt you.
No, that was an important point. And I think what will happen over the next, months to years is really a lot of other labs trying to find this clonotype in their databases that, if they're not public, they have them within their lab and trying to understand in their patient populations if this cell type is there, if this clonotype is there. So, Yep. So next thing they did is they made a protein, and they wanted that was it was specific for the sequence of the clonotype, and they wanted to do really modeling to show, can this protein bind to the MHC, and they compared that to the superagonist, which I mentioned before, binds the MHC at a really 1,000 times better than the native form of insulin. And they also did the same XID protein from a healthy control. And so what they're using here, they call it molecular dynamic simulations, and it's a biocomputational way to model if these different peptides are stabilized within the MHC2 loading site. And when they do this, they really find that the one that binds the best is their X idiotype. That's from the CDR3 region of their clonotype, and actually binds better than the superagonist. And they go into different things about the specific binding, and they talk about there's just basically more fluctuations in the binding of the superagonist, and these particular aromatic residues on the X idiotype peptide really contribute to a more favorable binding. And it goes back to kind of what we discussed of how, changes in insulin can change its conformation, that changes the binding. And I think that was important for them to prove, because they wanted to show that, the sequence that this clonotype recognizes is a peptide that binds to MHC, which would be the start of a T cell reaction that could cause autoimmunity. So that was important for them to find out. The next thing is really then using that peptide to see if it stimulates T cells. And they did that in a couple different ways. They were able to see if just the stable peptide bound to the MHC if you have it, the full sequence compared to a truncated sequence, if it was able to bind compared to the mimotope, which you talked about, Vincent, or the native form of insulin. And again, their peptide that was sequenced from the CDR3 was able to bind really well, but not the healthy control and not the native insulin. And that was able to then also stimulate CD4 T cells. So I think the story they're building here is that this peptide does in fact stimulate cells by binding to the TCR, and they also use a blocking antibody as well, and that inhibited proliferation. So I think that, this is proving that this peptide, wherever it is, if it's in people or if it's not, but it is able to stimulate the cells necessary to cause autoimmunity. So the last, let me see here. Okay, yeah. So the next couple things they do is they wanted to generate a cell line, and they were able to do that. They took the dual expressors, they immortalized them by using Epstein-Barr virus, which you can do to produce these continuously growing lymphoblastoid cells. And then from that cell line, then you can produce monoclonal antibodies, and those monoclonal antibodies you can test. One thing that's interesting, they have this immortalized cell line. What a great way then to study more functionality of BCR and TCR receptors, which I would love if we're really trying to show that this is like a unique developmental lymphocyte. They don't really do functional studies, which I would have loved, but they do confirm that this immortalized cell type, it does have the BCR, it does have the TCR. They're using single cell analysis to show all this. It also expresses the same surface markers. And the monoclonal antibodies that are produced from that are able to stimulate CD4 T cells. I'm going to go into the last kind of section of this paper with one thing I totally understand and one thing I don't understand, and maybe Cindy can help me with this. So after the experiments where they use the monoclonal antibodies, they use the monoclonals in what they consider the natural form, but they also produce it recombinantly. Both of them are able to stimulate CD4s from type 1 diabetic subjects. And they also make tetramers, which we've talked about a little bit before, but a tetramer is essentially like a soluble MHC, and it can bind your peptide of interest. And on the other side, you put a biotin molecule. And you can take 4 of these complexes and then enzymatically bind it to a streptavidin molecule. So now you have a nice molecule that can probe your T cells, and then you can detect it by fluorescence. And so by using this, they were able to, and they use the same, they use the same the DQ8 MHC molecule, which is relevant to their study. And they again co-cultured PBMCs because they can't isolate these cells. There's too few of them for 7 days, and analyzed the culture and found the presence of these tetramer positive CD4 T cells. So showing that this peptide binds to CD4 T cells by the tetramer from using the peptide of interest. And lastly, they kind of show that the peptide that they produce plus the mimotope, which would be kind of the enhanced version of insulin, stimulate kind of similar populations of CD4 T cells. So they're trying to show that this peptide maybe is driving type 1 diabetes. So the last thing that they do is and Cindy, it's this part was really challenging to understand, but I think what they're suggesting here is that a cell can present the variable region of its antibody to a CD4 T cell and stimulate it to respond. Because I think we normally think of presenting antigens, different peptides from either a self-antigen or a pathogenic. But what they're suggesting here is that if you give the monoclonal to a population of PBMCs, that there's going to be presentation of the variable region, which can stimulate the CD4 T cells. I thought that was kind of a convoluted experiment, and I don't think I quite understand it, but it's basically suggesting that there's similar, that the CD4 T cells are going to bind the sequence on the monoclonal which they produced, which is specific to their clonotype.
So I think it's this mimetope idea, right?
Yes.
Where if you have that you have this weird peptide that comes from the CDR3 presented in the MHC, and that then binds to the TCR, then the antibody that contains that CDR3 will also bind to that TCR.
Yeah, exactly. That's right. So yeah.
But did they add
that's the mimetope part of it. So what they're saying is that because of the fact that this particular peptide can be presented in this risk allele MHC, it itself looks, because it's part of an antibody, but that part of the antibody then binds to the TCR. So that original antibody will also bind to that TCR.
That's right.
Right.
So what may be weird for some listeners is that BCRs on a cell surface, it can also be released
soluble
as an antibody, but it's also going to be chopped up by the proteasome and put on MHC, just like any other cell protein, right? So that's exactly, it can work either way.
Sense, yeah. And then when they were able to block that by blocking the MHC, and then it does not work. So I think that last experiment was to show that the specificity of this particular variable region of the antibody.
So can you look for those antibodies in people, right?
Well, yeah. I mean, yeah, you can. Hopefully more than 3 people, which you described was what they had kind of ran it against for the BCR. But yeah, for just looking for antibodies, I think that would be their next step, and a lot of other groups next step.
Because if it's pathogenic, then you should have a good amount of these antibodies around, right? It's not going to be rare. Although, the rarity of the cells is just so weird, and it's causing this massive disease. I don't know about that.
It's only a couple cells, like 4 cells or 7 cells.
I don't know if that's the total complement in these patients. It's got to be more than 3 or 4 cells, but maybe that's all they were able to get. I mean, they have a limited amount of blood they can get from these patients, right? So
right.
Maybe it's 100 mils at the most, so you still don't have a lot of cells there.
Yeah.
By the way, Steph, so the 37 million BCR sequences was from 3 healthy patients. They also did similar numbers from type 1 diabetes patients, and they had a few more. They had 18 diabetic patients, but still it's not enough to be comprehensive, right?
Right. Yeah. So, I think this paper in general, when we're looking at kind of just like the big picture, they're looking at this rare B cell subtype that does express TCR, the CDR3 region of the heavy chain gene in these dual expressors and codes an antigen that does bind to the risk allele, the DQ8. And then when you synthesize that peptide, it binds to the MHC. It can stimulate CD4 T cells, most in type 1 diabetic patients, but not in the healthy controls. And then you can produce a cell line that makes the antibodies that also stimulate CD4 T cells. So I think they did a good job of showing us that this could be potentially a public clonotype. It's just, can we find it in people? And the other thing, can it really drive autoimmunity if it's such a rare population of cells? And lastly, they do suggest that this shows that compartmentalization of these B and T lymphocytes is not absolute, and this may violate a paradigm. And I would argue that this probably is just like many other autoimmune diseases and lymphomas, is just an aberrant cell type that expresses TCRs and BCRs, but it actually also expresses a lot of other immune cell transcripts as well. And so it's unique to study. I think it's good to study. I don't think it's paradigm shifting, but more studies will come out. We'll see if that's true. So overall, what did you all think about the paper?
Well, go ahead, Cindy.
I was going to say, I went into it with a hefty dose of skepticism, as I'm sure almost all immunologists did, because of exactly that. They're very clear in their introduction and in their conclusion saying that, the specific compartmentalization, T cells or T cells, B cells or B cells, is not absolute, and this is violated by what we found.
And that's really that's really what the media picked up on too.
And it's the media picked up on it, and everybody's hot on this because it really totally, completely changes how we think about the immune system. And I don't know that they could go as far as they did in saying that. But I do think that it is very cool that you can have a chunk of an antibody that can be presented and act like a T cell receptor antigen, and then that same antibody can turn around and bind that T cell and activate that T cell. So you have this potential to kind of like have this accidental antigen that can basically trigger T cells in a weird sort of way with antibodies and drive autoimmune responses. I think that part of it is really cool and kind of gets covered up in the showmanship of trying to try and say that this is just a totally new way of thinking about, the immune system development. So I think we can stick to T cells are T cells, B cells are B cells. This is a really weird, unusual thing that happens specifically with a subset of risk alleles for this particular disease, and that more studies will have to be done to see whether there are other cells expressing both T cell receptor and B cell receptor. And I think you raised the most important point, Steph, is that they ignore the fact that they do express other cell markers. And so it could simply be that this particular cell type, for whatever reason, has some epigenetic changes that allows it then to express multiple lineage receptors, and have, it itself, yes, it violated the paradigm, but it's because it's an aberrant cell type in this particular disease.
Yeah, I agree. Because in the paper, I think this is the first, like what you said, Cindy, this is it's the first time that's been discovered where they can take this clonotype that encodes for an antigen that stimulates it. I mean, that's novel and really exciting. And I think they do prove that to us. But you're right, it does get covered up a bit with the showmanship of it. And I get it. I mean, it is interesting, but stain those suckers for some mast cell markers or NK cell markers on the surface if we're going to, if you see the transcripts, like look at the surface and see if they're there.
Yeah. Yeah.
So I think I had less bias coming in, probably just from ignorance, but I think this is just a weird cell. Yeah. Like you guys are saying, it's weird, and anything can happen, right? Just because we say that this only T cells have TCR and B cells have BCR, it doesn't mean that you can't have a weird cell that has both, but has lots of other things as well. What I think is quite clear is that this mimetope can definitely stimulate the T cells either as a peptide or an antibody, and that's probably the relevant part. The question is it at substantial levels or enough levels that it would cause disease? Right. And I'm not sure that it is. Otherwise, we might have seen it already. So now people can look for it. I think in people with type 1 diabetes, you have to find either these antibodies or the peptide doing this. Otherwise, it's just a coincidence. It seems like it would be a weird coincidence that this would happen, right? But
right. And the bummer really is you would want to isolate these cells. Well, first you'd want to find them in mice. So you could do like adoptive transfer studies. This is cause type 1 diabetes, but I think the problem, I don't know, conveniently or not, is you just can't isolate enough to do more robust studies. And so, yeah, I think this will be heavily, investigated to try to show if it's true or not. And
so they say our findings lay the groundwork for examining whether the X autoantigen could serve as a diabetes risk marker. And they basically, we have to look at a lot more patients to know this, because we've only looked at a handful. And I think that's probably ongoing, right? We'll see. Right.
So don't rewrite those textbooks yet.
I don't think you have to, because as Cindy said, the rules are still there, but shit happens, right?
Yeah.
Things happen, things screw up. We know that. And so we shouldn't be surprised that there are a couple of rare cells that I'm particularly interested in what developmental switch got messed up to let this happen, right?
Yeah.
I'm not sure you could ever reconstruct it, but it would be interesting to know. But how far back in the development of this cell did this thing happen? So that would be cool to sort out.
Totally. Well, thanks for coming along with this paper with me. And, a lot of it, most of it, I was able to get through. It's also nice, I think, for listeners to know that helping each other work through some of these more challenging experiments. And glad we got to do it.
All right. You guys want to just do some picks since we're a little long here?
Sure.
Skip some emails. Cindy, you don't have a pick. Are you okay?
I I didn't have a pick, and actually the fire alarm is going off here.
Yeah. You need to
I
leave.
don't know if you can hear it.
I do hear it.
I'm gonna sit here and pretend it's not happening because I have my screen down. So
we don't want
do what I do, right?
Well, we could either do,
I didn't have time to get a pick this week. Sorry.
We could either do a couple emails or picks. What do you think?
You can do your picks.
Okay.
That's fine. Yeah.
So is this going to annoy you though? Because I don't know if I should.
No,
I can't.
Don't worry.
No.
Okay, I don't know if you can keep hearing it. I'm
loud.
trying to cover up my mic so you don't hear
fine.
it.
I'll cut it out, obviously.
Okay.
Okay, so there is a new initiative by a friend of mine and also a scientist in virology. We've talked about him before, but Ephra is somebody who is really passionate about science communication and has started this thing called Unique Scientists. And I'm submitting this as my pick of the week. And basically, you can submit yourself as a unique scientist and talk about what you do and your journey and how you got to science, and really try to showcase the really vast amount of people doing what we do and what they do. And it's really very cool and very exciting. So just wanted to put that out there if you were interested in kind of highlighting your journey in science, whether you think it's unique or not. I bet other people do and would really like to learn from you.
That's pretty cool.
Yeah.
So I came up with one too. Yeah, I love that.
It's neat.
Oh, you came up with one. Good.
Yeah, I did. I did come up with one. But yeah, I saw his thing on Twitter, and I thought that was really neat because I think that shared experience, is really important to hear other people's stories.
Yeah,
Definitely.
the more people hear about scientists, the better, because people just don't know what we do or what we're like.
Yeah,
so
definitely.
it should all be out there. What did you end up with, Cindy?
Oh yeah, I remembered that a friend of mine told me about this the other day. So there's an app that's called Researcher, and I don't know if you've heard about this, but so the Researcher app is the way I heard it referred to is Tinder for scientists. So you put in a whole bunch of preferences of topics that you like, and it basically works like an aggregator for papers, and it pulls up the papers that are close to the things that are in your interest area, and you can read the titles and you can read the abstracts, and you can swipe right and swipe left if you like it or you don't like it. And it learns your preferences. And so you can use it to kind of, when you're sitting around and you want to see what new papers have come out and in your area of interest, you can use this app and it shows you that. And then you can go to the full text if you have the subscriptions.
It's interesting.
Yeah, I thought that was neat.
So I use Feedly to do this similar thing. Feedly.
Oh, okay. No, I didn't
you
know
can
that one.
put in the RSS feeds of journals into Feedly, and then it will download all the articles or the titles and so forth, and you can go through and save them. And that's how I identify papers for the podcast. Usually I go through Feedly, and you can do it on your phone and you can do it anywhere. So if you're waiting for something, you could look at the literature. But this is a similar thing. It's cool.
Yeah. Yeah.
Because most people don't want to deal with RSS feeds. What's RSS? So my pick is a podcast called Stellar Conversations. Stellar, STLR, stands for Science and Technology Law Review. It's the Columbia Science and Technology Law Review. And, this is, you know what the law review is. It's a bunch of law students who, study legal issues and so forth. And this is just extended to science and technology together. And so they have a podcast called Stellar Podcast. And it's on episode 3, but I want to point out the 3rd episode, which is called Compulsory Vaccine Laws, with Vincent Racaniello and Erwin Chemerinsky.
Cool.
And so Erwin Chemerinsky is a lawyer who might be a judge, I'm not sure. And they ask him about the legality of vaccine laws. It's very interesting. And with me, they talked about the science, so they have a little podcast with both in it. So he was interested. I really enjoyed listening to his part because he was clear that all the vaccine laws are completely constitutional and legal. There's no doubt about it. Of course, if this is a judge, we do have someone here in this country who would just say, oh, it's a bad judge, right?
Yeah.
For expressing those opinions. But anyway, it's a cool podcast, so check that out.
Very cool.
Stellar Conversation. So the funny thing is, when they talk to you at the end, they go, "Thank you for this stellar conversation," right? Because it's the podcast name, right?
I like that.
So they said that to the judge, and he said, "Oh, you're very kind." Because he thought he was, you get it.
Yeah. That's great.
All right. That is, go ahead. What were you going to say?
I was going to say one quick thing. I wanted to give Efra full credit by saying his last name as well. So it's Efra Rivera Serrano. So just to put that out there.
Yep. All right. That's Immune 21, where we try and spread science in a different way, podcasting. There's all kinds of ways you can spread science. They're all good. You can find it at microbe.tv/immune. Please subscribe. You can subscribe on your phone or tablet, through your podcast app. Please subscribe. Really helps us to know how many people are out there. And if you really like what we do, consider supporting us. It doesn't cost a lot. You could give as little as $1 a month. That's nothing. It's nothing. It's not even a cup of coffee. It's not even a subway ride here in New York City. I don't know what you can get for $1 anymore. Can you get like chewing gum? Not even.
Nope.
Can't get anything for $1. In fact, if someone wants you to give them some money, and you give them $1, they look at it. Like, on the subway, people often ask for money. If you give them $1, they look at it and say, "What?" But we are happy to take your $1 because there's so many of you, it would make a difference for our expenses. So you go to microbe.tv/contribute. And $1 a month, you get not only Immune, but TWIV and TWIP and TWIM and TWEVO. I mean, it's all cool science by cool scientists. And so please help us out. And if you give $1, we'll answer your email first. That's a good perk.
Yeah. Right to the top.
Right to the
Except
top.
Except for when we choose a paper that's so complicated we didn't. But we will next time.
True.
Yeah, we do have emails here, and
we do.
Sorry we didn't get to them, but we don't want to keep your ears burning for much longer. Cindy Leifer's at Cornell University. Cindy Leifer on Twitter. Thanks, Cindy.
Thank you.
Steph Langel's at Duke University. Stephanie Langle on Twitter. Thanks, Steph.
Thank you very much.
You were very brave to do that paper.
Woo! Yes. Well, I feel exhausted. My adrenaline's coming down now.
I mean, your show notes are amazing. It's like a thesis.
Yeah, I know. But I was like, I really don't want to screw this up. But hey, it's a learning process. And
it is, for sure.
I love it. I love doing it with you guys. I know we're at the end, but thank you for really including me, even though I'm, a younger scientist in training. I just really appreciate you both.
Thank you for including me, even though I'm an old guy. It can go all kinds of ways. It's all good.
Yep.
I'm Vincent Racaniello. You can find me at virology.ws on Twitter, Prof. VRR. Music on Immune by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.